Okay, as this loads up, this is my take two on the protein synthesis. I had to redo the notes a little bit towards the end. It sort of messed me up. But you will never know how because I will not tell you. Anyway, the title of these notes are protein synthesis. We started doing these in class. So that's the title. You should have a Cornell note paper. And the essential question is how are proteins formed? So from these notes, you will learn how we get the information from DNA, that sequence of DNA we call a gene, and how it turns into a protein, and then hence a trait. So here we go. All right, so this is a short little video, and I showed this in class, and I think it's fascinating. Just so you can see the process. This is an animation, of course, of a protein being formed. So that little red thing is an amino acid, and we're going to learn that an amino acid is a building block for a protein. That big blue thing or purple thing is the ribosome that resides in the cytoplasm. The yellow strip is RNA, specifically mRNA, which we'll talk about. It has nucleotides just like DNA, and that's where it actually gets its information from. And the big green triangles are what we call transfer RNA. They're bringing the amino acids, they're the amino acid carriers, and they're bringing them to the ribosome. And they drop them off and they build that red string coming out is the amino acids that are hooking together, and eventually they will share. I mean, they'll eventually make a protein. So that's the little process, like a little factory. I think it's sort of cool. All right. If you, like I said in class, you could probably maybe feel it inside you. No, you can't, because it's your body is doing a billion things right now that you have no idea what is going on. Anyway, we usually spend a lot more time on this section, but it's just going to be one slide. It's just the functions of proteins. We think about proteins and mostly a dietary need, not so much how it works in your body or what kinds of things it does in our body. So here's a list about 6 of them that the most common. So structural things like keratin and collagen, you'd find that in your hair, nails, anything is that sort of elastic, like your cartilage in your nose, your tendons, ligaments, things like that. Hormonal proteins, those are chemical messengers. So those carry chemicals to certain parts of the body that need it, right? Um, Storage proteins, ovalbumin and casein. You notice that you see egg and milk. So those are for developing organisms, right? So they're stored in areas where an organism may not necessarily have access to it, whether it's inside an egg or an infant, let's say a mammal who needs to breastfeed to get their protein. Transport proteins. Another type of a carrier protein. Instead of a chemical messenger, these are actually just proteins that um, things can bind to. For instance, hemoglobin is the protein that you find in red blood cells that carries oxygen from your lungs to your tissue. Movement proteins, these would be associated with your muscles. We have 2 types of proteins that are a slide back and forth. They contract when you flex your muscles, and they relax when you relax your muscles. Those are 2 different types of proteins in your muscles. And the one that we've talked about last unit when we talked about DNA helicase and DNA polymerase are enzymes. So they help break down or build things. They're chemical reactions, help, those chemical reactions that take place. So for instance, if you lactase breaks down the lactose in the milk so you can use it as glucose, where if you're intolerant, you may be deficient in that particular enzyme. And pepsin is the one that breaks down actually proteins. Okay, so what are proteins? As I mentioned before in that video, you had those little red blobs on the end of those green triangles. Those were amino acids, and you put a bunch of amino acids together and they form a protein. And that may sound familiar. We do the same thing with DNA. We have the nucleotides, and you put a bunch of nucleotides together and you make a DNA molecule, a huge molecule, right? So we talk about the monomers being the amino acids and the polymers being proteins. I know that I've mentioned that term before. Proteins are made up of amino acids connected together and with a different type of chemical bond, a specific bond just for proteins, and those are peptide bonds. The other two bonds that we talked about in DNA, we had the hydrogen bonds, the weak hydrogen bonds that are in between the nitrogen bases, and then we had the covalent bonds that are between the sugar and the phosphate. These peptide bonds are a little stronger. They need to be held together. And what happens, you put these amino acids together, and just based on the chemical, what's the term that I'm looking for? The polarity, right? It will form it into different shapes of the protein into different shapes. And that's where we're going to be, um, leads us to our next slide, right? So you see here, and this just a animation of, just a picture of amino acids. That's what these three letters. And so we have a string of amino acids. How many amino acids there are and what order are going to determine the shape of the protein, right? Now, the structure or the shape of the protein is going to determine its function. Um, you're going to hear this a lot if you take a lot of science classes, especially like if you're seniors and you take anatomy and physiology. I like to talk about it. I lead off every beginning of every unit. What's the structure of this system, and why is it shaped the way it is to help with its function? Simple example is cells, right? If you think about plant cells, and I'm looking outside at a tree, and the green leaves, they have cells that have chloroplasts in them because they photosynthesize and they have to make energy. Well, if I look at the trunk of the tree, it's still a plant cell, but it doesn't have chloroplast. And so you can see how the structure of each of those cells determine what it does, right? So the trunk has a lot more rigid cell wall, things like that. All right. So how does structure determine function? Well, we're going to talk about how the sequence of DNA, or if you remember that vocabulary word gene, remember that gene is a sequence of DNA that codes for a protein. So that sequence of DNA is going to determine how many amino acids are in that protein and what order those amino acids are in. Those two things are going to determine the structure and then hence determine the function. Okay, protein synthesis. How do we make proteins? We know that the DNA stores all the information, the genetic information that we need. So how does it get to the point where it can, make a specific protein? We know that DNA cannot leave the nucleus. So what happens is it transcribes it into what we call mRNA. And we're going to compare RNA and DNA in a minute. But that mRNA can leave the nucleus. So that mRNA is able to leave the nucleus and go to the ribosome, that blue blob that we saw in the video. The yellow strip was the mRNA, and then the string of those red things being put together will develop into a protein, right? And it's called translation. So the first part, DNA to mRNA, is what we call transcription, and the process from mRNA to protein is called translation. One of the things I do want you to be careful with, do not say, and you underline this really, is that DNA does not turn into mRNA. DNA, remember, needs to stay DNA. All right. It cannot leave the nucleus. It's always the DNA. Remember that it needs to make copies of itself exactly so it can make it to new cells, so those new cells can make the right proteins to make us who we are. So it's transcribed. So mRNA is transcribed from DNA, and the proteins are described from mRNA. mRNA is recycled though. We always make new mRNA. So that's transcription first, translation second. I always remember that it is in alphabetical order, with the transcription is alphabetically before translation. So it always comes first. All right, so these are based on what we talked about before. I'm not going to spend a lot of time on this slide because we know that how important DNA is. And we talked about quite a bit how the order of the bases determines the order of the amino acids in the protein. And we'll learn how we're going to determine how that order in sequence or the order and number is determined. Again, another review slide. I should have changed this. I like sequence instead of portions. I like because it is a sequence. So genes are sequences or a sequence of DNA that code for specific proteins. And we'll learn that in a minute. All right, flow of information again. So before we go into the actual protein synthesis, we thought it would be important at this time to differentiate between what DNA is and what RNA is. They're both sure, they have the same, well, let me just start. Okay, so we know that DNA starts with deoxy, it stands for deoxyribonucleic acid. RNA is ribonucleic acid. I believe I told you when we were talking about deoxyribonucleic acid, is the deoxy is that has one less oxygen than a ribose, right? So here's your deoxyribose and here's your ribose. And it's pretty amazing to think that one oxygen atom can change the molecular structure of something so great, right? Let's see, number of strands. Remember we talked about DNA being a double helix. So it is a twisted ladder, where RNA, you saw in that little yellow thing in the little video, was just a single strand. This is where it's going to get a little bit confusing, especially when we go back and forth from DNA and RNA. You can see that instead of thymine, I have uracil. Okay, so that's the only thing that's different. So we still have adenine, we still have guanine, we still have cytosine, but with thymine, it is replaced by uracil. So we have an adenine here, cytosine and guanine, but now we have a uracil. So whenever we ask you to, write the complementary strand of a DNA molecule, to make it a DNA, make sure you see a T. If we tell you what would the mRNA strand be, you want to see a U in there, right? Location of the cell, we talked about this. DNA stuck in the nucleus. RNA can move back from the nucleus. The mRNA specifically can go from the nucleus to the ribosome. And then we have the other different types of RNA that's residing near the ribosome. And this is that next slide. What are the three types of RNA? Messenger RNA, we've seen that before. So that takes the message from the DNA to the ribosomes, where the proteins are made. The transfer RNA brings that specific amino acid to the ribosome. So that big green triangle in that video, that was transfer RNA. Ribosomal RNA is actually just makes up the ribosome where the proteins are made from the amino acids. We don't normally use in a normal explanation that we usually just call it ribosomes. We'll hardly ever hear us refer to it as ribosomal RNA, but we will mention messenger RNA, and we always just call it mRNA, and then tRNA or transfer RNA. Those are the two that you're going to see more often. All right. Okay, base pairing rules. Really based on Chargaff as well. I think he found the same thing. All right, so just like in DNA, cytosine pairs with guanine. So anytime you see a G, it goes to a C. Anytime you see a C, it goes to a G. Adenine pairs with uracil. So this one's a little bit tricky because if you see an A on DNA, it's going to be U. If you see a T on DNA, it still will be an A, right? So, but if you go from RNA to RNA, which we do sometimes, the U will be an A and the A will be a U. Okay, so for example, if you see our DNA strand this, I'll give you you can pause the video and fill it out and see if you get it right, but you should see the U's, right? So we have the AUG, and then we have a UU down here, right? So everything else is the same. Okay, so the process of transcription is coding DNA into an mRNA strand in the nucleus, right? So you can draw a picture, and it doesn't have to be this exact, but what we want you to understand is that you have, so we can't see the nucleus is huge, right? You have to imagine like this is like a tiny part of the nucleus, because this is the enzyme. We saw DNA polymerase. RNA polymerase is the same thing. It builds that RNA molecule. You can see these nucleotides being added on to the strand of the exposed DNA, just like in replication, although we're making an mRNA, right? So it says draw a picture. So it could just be a rough, and just make sure that you show the DNA, that it's closed, and then it's open during that transcription process, but then it can close back up again, right? And you're just making some type of a RNA molecule. So it's actually going on this way, right? This is newly made, and these guys are getting made, and it's opening up this way, right? So, okay, these have already been made. Okay. All right. Transcription. So this is the process. Oh, this is the same thing. Sorry, I thought. So this is just transcribe the sequence. And again, you can pause the video and see how you do. All right. Again, we have some use. All right. Translation. So this is the process in which the mRNA strand is coded into amino acids. And this is changing the language. And that's where the term translation comes. So you can see, and again, you can draw a picture. So this is the mRNA, right? And this was that yellow strip that you saw. This is those green triangles with the red dots on them. So these are the tRNAs dropping off the amino acids and then building your amino acid sequence. This tRNA, it can go back. It dropped off its amino acid. It can go get another one. So it's pretty amazing that how that happens. And we do have base pairing rules, and that's how the tRNA knows that it needs to drop it off, because it matches up with the mRNA, right? So this sequence, see this yellow guy is A, and this orange guy is U. So this A is going to match up with this U. And then it has these two G's, which are the teal color. It's going to match up with these two blues, the dark blue colors of C, right? And it's going to drop off its whatever amino acid that is. All right. And you can see another aspect of it. We are going to learn that we use this mRNA sequence to determine what amino acid is going to be dropped off. All right, so we have the mRNA with the corresponding tRNA letters. So how they specifically match up is going to determine a specific amino acid, right? And we know all that. It's pretty fascinating that they found that out. So we're going to call, and I think last time in our warm-up, when we did the warm-up, what's the DNA, what's a complementary strand for this DNA template? We had it divided into threes, and I told you to sort of, make a mental note of that. And this is why, because I'll go back here real quick. You can see that everything is divided into threes. The tRNA has 3 nucleotides on it. It's going to match up with the 3 nucleotides on the mRNA. So we are going to divide the mRNA strand into a segment of 3 nucleotides, and we're going to call those segment of 3 nucleotides a codon, one codon. So if you look at here, we have 1, 2, 3, 4 codons, right? Because there's 12 letters divided by 3, and that's 4. So one codon codes for one amino acid, and then remember that a protein is a string of those amino acids put together. So here, how do we read this chart? Well, you always, go from left to right, just like you're reading a word, I guess, right? So the big letters indicate the first letter of that series of 3. So the A starts there. The U is going to be the next letter going inside to out. And then the G is going to be the smallest letter. So A, U, G. And you look here and it says methionine. Some of these will be abbreviated and we don't care if you abbreviate it. So that would be a MET for abbreviation. So that's that amino acid that's being coded. Now, I'm going to go ahead and we'll talk about this a little bit later, but every protein needs to start, like if I'm going to make a protein, the sequence of amino acids has to start with methionine. That tells the cell or the polymerase, actually, the one that's building it, this is my starting point. If you don't see this AUG, don't start building the protein yet. Only when you see this AUG can you start building and putting the amino acids together. All right. So always remember, I'm just going to say some practice problems aren't always going to follow this rule. All right. So sometimes we might have you practice and it won't start with methionine, but we try the best we can. All right. So methionine is our start codon. All right. Our next is CUC. So maybe if you can, pause the video and see if you can find out what CUC is. And I know sometimes these words are hard to pronounce, but that's okay. So did you get leucine? CUC, leucine. All right. Okay, so next, what's next? AGU. What is AGU? Serine. All right, did you guys get serine, AGU? Now, one thing I do want to mention, and I'll just go ahead and briefly mention it. If you can see like valine over here, you can see that it has GUG, GUA, GUC, GUU. All of those sequences will code for the amino acid, right? We have only 20 amino acids, but there are 64 combinations. So there's a lot of repetition, right? So, you'll see that. I think leucine has 6, right? We have 4 over here that code for leucine, and then we have 2 up here that code for leucine. So, it's all. Okay, so the last one, UGA, and let's see if you get that one. And yes, that's stop, right? That's a stop codon. That tells, now it doesn't actually bring an amino acid, but it tells it, okay, we're done. Stop making the amino acid. The sequence of amino acids is going to go make the protein. And then that's it. And that's done. And it may start over again with a new series of mRNA. All right. So that yellow strip, and it could be shorter or it could be heck long, but it always needs to start with AUG and it always needs to stop with a stop codon. A amino acid sequence for a protein has to start with AUG and end with a stop codon, right? And that determines how long that protein is. And remember, structure determines function. We're not going to talk about mutations so much, but like if there's a missing letter within the sequence, then it might not hit a stop codon and go on forever, and it could cause a disease or some, it could be, most likely it's, won't cause any harm, hopefully, but sometimes it does, right? If it's even like we're talking about, I talked a little bit about hemophilia, or not hemophilia, but sickle cell is a change of one letter in the DNA sequence, and it makes a different protein, right? So it's all pretty amazing. All right, so these are just all practice problems. I guess this wasn't really a practice problem. It just gives you the answer, but this does, right? So go ahead and practice. See if you get the right amino acids. I don't know what we're gonna do, but we normally give you this chart. We'll probably talk a little bit about it amongst the teachers, how we can have you access this chart, because we certainly don't expect you to memorize it. Okay, so so go ahead, see if you get these, and then watch this Amoeba Sisters review. Oh, you know how much we love the Amoeba Sisters. So that's it for the notes, and we will see you next week on Thursday, Friday, because we have asynchronous days on Monday and Tuesday.